Hello and welcome to today's ISHR JMCC cardiovascular webinar. Thank you all for joining us. A few quick announcements before we get started. Today's webinar is by Sakthivel Sadayappan. His paper was selected as the JMCC editor's choice. A few quick notes. Just a reminder that JMCC has called for papers on special topics of interest. We have two submission windows. One is just about over, so if you're interested in submitting a paper on metabolism and inflammation or cardiac fibrosis, try to get those in by September. We have another window of submissions coming up for September to March. These special issues will focus on sarcomeric protein mutations and ion homeostasis and arrhythmias in the heart. So please check these out if you have interest in submitting original research to JMCC on these topics. Another quick reminder to check out JMCC's Scientist in the Spotlight podcast, which can be found on multiple platforms, including Podbean, Apple Podcasts, and others. You can just search Scientist in the Spotlight JMCC and you'll find the most recent episodes. So with that, I will turn our program over to today's chair, Professor Sam Harris at the University of Arizona, to introduce our speaker. Please enter any questions into the Q&A box, and we'll have a Q&A session at the end of the webinar. Again, thank you for attending, and thanks to JMCC and ISHR for their continued support.
Thanks, Ron. Good morning, everyone. It's my pleasure today to introduce my very esteemed colleague and friend, Professor Sakthivel Sadayappan. Sakthi is a professor of internal medicine at the University of Cincinnati College of Medicine. He started his career, his PhD, in India, where he received his PhD from Madurai Kamaraj University in 1999. And from during that time, he had defined his interest very early on in cardiac hypertrophy. So the title of his thesis was Molecular Aspects of Cardiac Hypertrophy. So he's staying on point. Along the way, he also, because a PhD might not be enough for some people, like Sakthi, he, in his spare time, earned an MBA in 2009. And then after he got his PhD, he went on to do a postdoctoral research fellowship at the Max Planck Institute under a DAD fellowship that he received when he was a PhD student, and he was, it's an prestigious exchange program. So he went on to do his postdoc there where he had done a fellowship. After that, in June 2002, he took a postdoctoral position in the lab of Jeffrey Robbins at Cincinnati Children's Hospital. And looking back at the CV, thinking about when I first met Sakthi, it was probably around this time that I first met him, and we had common interests in our favorite protein, myosin binding protein C. So it was a little daunting to see that we have known each other nearly 20 years now, Sakthi. So what a pleasure. So from there, he spent time at Cincinnati and rose through the ranks for research associate and research instructor until he finally took an assistant professorship at Loyola University in Chicago. He was granted tenure and promoted to associate professor at Loyola in July 2016. But immediately thereafter, he was recruited to the University of Cincinnati College of Medicine, where he achieved the rank of professor and director of heart research, heart, lung, and vascular institute. So quite a rapid ascension for Sakthi. Now to kind of cut to the chase, he has over 137 publications. He's been an attentive and engaged mentor to numerous successful grad students and postdocs, and many of whom have now begun independent careers, and some of which are studying myosin binding protein C. The amount of research support is truly awe-inspiring that Sakthi has gleaned. So he has multiple R01s, and he has been a constant supporter of the AHA, and the AHA has been a constant supporter of Sakthi and his students. So it's really, really remarkable. And let's see, without further ado, I will let Sakthi tell you about our favorite protein. Thank you, Sakthi. Uh-oh, is your volume on? Unmute?
Okay, do you hear me? Yes. And there you see the full slide, right? Full screen.
Yes.
All right. All right. Sam, thank you so much for such a great introduction and also moderating the session. This is the topic of our career interest. So I'm really excited to have you on board with me today. So today's presentation is based on our recent study and the article that appeared in JMCC entitled Amino Terminus of Cardiac Myosin Binding Protein C Regulates Cardiac Contractility. I take this opportunity to thank the editor, Dr. Rong Tian, and the JMCC editorial board for selecting our article for the cover page, as well as the editor's choice. Also, I would like to give a very special thanks to Ron Vagnasi for doing such an extraordinary job organizing the JMCC-ISHR cardiovascular webinar and inviting me to present here today. Also, I take this opportunity to thank and acknowledge all my collaborators and co-authors who have significantly involved and contributed to the present study. Importantly, I would like to congratulate and thank my two former graduate students, Dr. Tom Lynch, who is now at LB Chicago, and Dr. Kumar, who is now at UC, who have done most of the work and shared the first authorship. These are my disclosures that I have nothing to do with my today's presentation. So what I will do the next 30 minutes, I will give a brief introduction or background about C protein and then present some of our earlier work on C protein proteolysis and then define the roles of the amino terminus of C protein that we reported in the JMCC latest article. So in adult human hearts, cardiomyocytes occupy approximately 75% of normal myocardial tissue volume. These are the cells that are responsible for generating contractile force in the intact heart. So if you look at this electron microscopic analysis, you see the sarcomere. Sarcomere is the functional unit of cardiac myocyte and contractility. So if you look at very closely the sarcomeres or sarcomere, it consists of both thick and thin filament proteins. Thick filament proteins are titin, myosin, light chains, and C protein. And thin filament proteins are troponin complex, alpha-tropomyosin, and actin. So my lab has been committed to investigate the regulatory, structural, and functional roles of C protein in health and disease, as included in this slide. So today we know contractile machinery is the downstream target of most of the signaling pathways, including kinases, phosphatases, and proteases. Almost every contractile protein undergoes post-translational modifications to alter cardiac function. So pharma companies are now highly interested to bypass all these signaling pathways and then directly target sarcomeric proteins so that the functional things can be changed or altered. So I'm here today to convince that C protein is one of the major key players regulating myocardial contractility in the sarcomere as well as in the myocardium. C protein was originally discovered by Dr. Offer, King's College of London. It was originally identified as impurity band C, as here you can see in the STS page analysis. And that's why it was named C protein. It's a contaminant when they prepared myosin total preparations. But if you want to blame who named C protein with a long name as cardiac myosin binding protein C, you should blame Dr. Fishman, who was at Cornell University Medical College. And also, if you guys are interested in taking any knowledge about C protein, I have a model quiz. You are very welcome to take it. I think there are nearly 50 questions. But you will see the answers at the end. So if you have any questions or comments, please contact me as well. It was one of the best momentums in my career to organize this one-day seminar when I was at Loyola University of Chicago, including Dr. Fishman, with all other world leaders who have been studying C protein. So coming to the C protein, there are three different paralogs of C proteins: cardiac, fast skeletal myosin binding protein C, and then slow skeletal myosin binding protein C. So these paralogs are encoded by separate genes. That's what we call them paralogs. They are not isoforms with a distinct expression profile in striated muscles. So in mammals, cardiac is limited to the heart, and compared to the skeletal paralogs, cardiac has a unique C0 domain and a phosphorylation domain with unique phosphorylation sites and a unique insertion within the C5 domain. So why we study C protein or cardiac myosin binding protein C? It is a transfilament protein connecting one side the thin filaments, another side the thick filaments, both the titin and myosin. And at the end terminus, you can see here it's connecting to myosin S2 as well as actin. And these interactions are dynamically regulated by phosphorylation. We also call C protein as a hard brake because it can control the level of octomyosin interactions. C protein contributes 2% of the total myofibrillar mass. Interestingly, C protein is localized to the cross bridge containing Keithjones of the A band in the sarcomere. And so if you look at in this schematic diagram, C protein is nearly 7 to 9 stripes at 43 nanometer intervals in the C zone. Very defined structural localization of C protein. So that's a very unique. There are 3 different knockout mouse models available for C protein, including Dr. Harris. She reported in 2012 in Sarc research. And then Dr. Seidman's lab, and then Dr. Lucy Carey, she reported one in 2004 in cardiovascular research. All these models tell us that compared to the wild type and heterozygous knockout mice, homozygous C protein knockout showed pronounced hypertrophic phenotype with wall thickening and chamber dilation. So clearly suggesting that C protein is essential for normal cardiac function. Also, mutation in C protein causes hypertrophic cardiomyopathy, which account for greater than 40% of all hypertrophic cardiomyopathy mutations. And these were first discovered in 1995 by 2 different groups. So one is from Harvard, Dr. Seidman's group. Another one is from Paris, Dr. Katie Swartz group. And after these 2 papers, researchers switched their focus studying from skeletal paralogs to cardiac paralog of C protein. And also C protein undergoes significant post-translation modifications that either directly or indirectly regulate cardiac contractility. So this is the domain structures of cardiac C protein, which is a 150 kDa protein. It contains 8 immunoglobulin domain and 3 fibronectin repetitive domains. So the various binding sites of a C protein with other proteins are identified here. And also C protein is a substrate for various kinases and calpain proteases. And not only for phosphorylation, but also C protein can undergo other post-translation modifications such as acetylation, glutathionylation, gluconylation, and citronylation. So altogether, a C protein is a regulatory protein and a central target of sarcomeric signaling. So based on our the literature's in vitro studies, C protein interacts with the myosin S2 region at its N-terminal region in the absence of phosphorylation in the M domain. So when it's become phosphorylated, this interaction is completely abolished and a change in both thick and thin filament orientation as well as contractile mechanics occur. However, it's unknown the mechanism of N-terminal region of C protein and then its regulation on cardiac contractility. So to determine the specific differences among these paralogs at the end of their N-terminus, Brian Lynn, my former graduate student who is now at Johns Hopkins, and I just came to know he recently received a fundable score on his K99. So he used the recombinant protein of N-terminal region of these three paralogs and then used them to permeabilize skinned papillary fibers at a higher calcium concentration and then measure the force of contraction as well as calcium, myofilament calcium sensitivity, and then the redevelopment. Data clearly showed that both fast and cardiac fragments increased force protection, calcium sensitivity of myofilament, and then the rate of tension redevelopment compared to the slow myosin binding protein C. So these data were concluding that functional differences and regulatory mechanisms among these paralogs are unique, and there are some comparable, but there are some uniqueness among these paralogs. So the clinical relevance of studying C protein is that C protein phosphorylation is significantly associated with several diseases in human, including atrial fibrillation, heart failure, cardiomyopathies, and even aging. So when I was a postdoc, my postdoctoral research were to demonstrate the C protein phosphorylation is necessary and sufficient for normal cardiac function. So to do that, we generated two different mouse models. One is the phosphoablation, another one is the phosphomimetic. And comparing to these controls, we were able to show that phosphorylation is necessary for normal cardiac function. Furthermore, others, and we also determined that C protein undergo proteolysis at its N-terminal region during ischemic reperfusion injury in both dog and mouse models. So these are the Western blot using the total lysate from the heart and then show that the N-terminal fragmentations. Using in vitro assays, again, my former postdoctoral fellow, Dr. Suresh Govindan, who is now in India and leading research in a private research foundation, he first demonstrated that C protein undergo proteolysis and the release of 40 kDa fragment, which is the predominant N-terminal fragment of C protein in a time-dependent manner. He also showed that the tissue C protein phosphorylation levels at these three different phosphorylation sites were significantly reduced, which is directly proportional to the release of C protein from sarcomere and increased 40 kDa fragmentation. He then showed that the release of 40 kDa fragment is very specific to the ischemic left ventricular anterior wall region using samples from rat myocardial samples using N-terminal specific myosin binding protein C antibodies. So one possible explanation that is during ischemic injury, calpain is activated that can degrade C protein like other sarcomeric proteins like troponins. So therefore, the question was, is calpain cleavage involved in C protein degradation and the release of the N-terminal fragments? So my former postdoc, Dietrich Kuster, who is now at VU Medical Center, Amsterdam, leading research on inherited cardiomyopathies, he used in vitro assays. So what he did was he used total myofilament proteins from wild-type mouse hearts that were incubated with the microcalpain at different time point or a different concentration, and performed Western blot against C protein antibodies and showed that calpain can cleave C protein and into several fragments, and then the 40 kDa is one of the predominant N-terminal fragment. So we then purified the 40 kDa fragment to determine the site of cleavage using mass spec. Strikingly, we determined that the 40 kDa was cleaved within the phosphorylation motif and named as C0C1F. The F refers the first 17 residues of the M domain. So the number of amino acids are 273. It's kind of closer to 29 kDa, but it runs differently on the STS page. That's what we call them as 40 kDa, but originally, in terms of molecular weight, it is 29 kDa. So Dr. Previous and Dr. Washers in their science article reported that the time-dependent release of 40 kDa or the C0C1F fragment, which occurred very early on in the proteolysis, and also they determined exactly the cleavage sites, which is in the phosphorylation motif. In the same group from Dr. Washa, so they performed in vitro motility assay using the various recombinant proteins of the N-terminal region of C protein, and then determined that the C0C1F is sufficient to inhibit actin sliding velocity, similar to C0C2 or the full-length myosin binding protein C, compared to the controls C0C1 or C0C1F mutated. The F regions were mutated to different amino acids, and they were not able to have the similar binding with the actins. Then we also used adult rabbit ventricular cardiomyocytes that were infected with adenoviruses expressing either full-length or 110 kDa, lacking C0C1F, or only the C0C1F fragments, and measured contractile properties. And then we determined that there is a significant reduction when we overexpress the 40 kDa fragment, including reduction in length shortening, relaxation, and contraction velocities. At the same time, Robbins' lab at the Cincinnati Children's, they also showed that overexpression of 40 kDa fragment in the heart, in the mouse heart, transgenically causes severe heart failure. We also used normal human skinned myofibrils that were permeabilized with human 40 kDa fragments, and demonstrated that the presence of 40 kDa fragment in the sarcomere reduces contractility. So clearly the C0C1F compared to C0C2 region includes critical binding sites, but it affects the one of the phosphorylation sites, 273, and also the absence of the rest of the M domain and C2 region. So altogether, from our own study, from others in the literature, both in vitro and in vivo studies have established the rationale for studying the impact of C0C1F region on cardiac contractility and cardioprotection. Even the 40 kDa fragment is toxic to cardiomyocytes, and we also showed that the C0C1 can activate pro-inflammatory responses. So then David Bearfield, my former graduate student, he is now at Loyola University of Chicago having his own research, hypothesized that ablation of the calpain targeted site in the C protein is cardioprotective during ischemic reperfusion injury. So to do that, we used a transgenic mouse model expressing C protein in which the calpain targeted site was ablated completely and expressed the mutant protein under or into the C protein null background. So the transgenic mice were conclusively shown to possess a normal basic structure and function. They are completely normal. They rescued like a wild type, the null background, and then shown up to 3 months by echocardiogram and hemodynamics. In collaboration with Dr. Keithjones at Loyola University of Chicago, we were able to show that ablation of the calpain targeted site in C protein, which can prevent the release of C0C1F, is cardioprotective during ischemic reperfusion injury by having significant reduction in infarct region compared to the control experiment. So now we are coming to the current paper, the paper that just presented or appeared in JMCC. So based on all this data, we hypothesized that the N-terminal C0C1F region of C protein is vital for regulating cardiac function in vivo, and expected that if we remove that C0C1F, and we can see a contractile dysfunction at the organ or the myofilament level. To achieve that, we generated a cardiac specific transgenic mouse in which the C protein was removed, this N-terminal region C0C1F, and then using alpha myosin heavy chain promoter, we overexpressed. In just overexpression without the null background, we were able to achieve almost 80% of total C protein with the truncation, N-terminal truncation. Only the 20% of the endogenous had the full-length myosin binding protein C. So which was significantly enough to study the functional impact of the N-terminal region, C0C1F. So we also confirmed by immunofluorescence analysis that the mutant proteins are properly localized within the sarcomere using the MIG-tag antibody. So MIG-tag, we had a tag at the N-terminal region of the C protein that can differentiate the endogenous versus the transgenic protein. We then determined that isolated hearts from the transgenic mice were really appreciably or significantly higher than the non-transgenic hearts. And also the transgenic mice showed decreased fractional shortening as well as presence of cardiac enlargement with the dilation, so compared to the controls. And then also in collaboration with Dr. Gilbert, we assessed the myoarchitecture in the exercised whole heart and showed that a normal distribution of myofibrils, like helix angles across the ventricular wall. This is kind of confirmation that ablation of C0C1 in C protein did not affect the heart structurally. We then performed electron microscopic analysis in collaboration with Dr. Roger Craig and Dr. Lee, and then demonstrated that the cardiac sections and negatively stained myofibrils showed normal sarcomere ultrastructure in the transgenic myofibrils. However, there were kind of weakness in the C zone region of the transgenic compared to the non-transgenic fibers. So then what we did was an interesting experiment. This kind of first experiment, we used various myosin binding protein C specific domain antibodies and to determine the importance of N-terminal region of C protein by permeabilizing them, and then measure their the force, P calcium force generation at the maximal P calcium 4 concentration. So here we used antibodies against the N-terminal region C0 or the C0 to C2 or C5, C8 or C10. So you can see here the maximal force only when we used the antibodies against C0 were significantly reduced compared to the other domains, suggesting that N-terminal region of C protein plays a role in regulating contractile force. In parallel experiment, we determined that incubation with the antibodies against C0 domain of C protein that had no effect because we removed the C0 domain in the transgenic myofibrils. compared to the non-transgenic myofibrils. So again, confirming the specificity and the importance role of the N-terminal region in maintaining cardiac contractile function. Next, James, my former postdoc, he's now back in Australia. He measured the proportion of myosin heads in the super relaxed state in the transgenic compared to non-transgenic skinned cardiac muscle fibers. And we observed that there were no significant difference in the proportion of myosin SRX state compared to the non-transgenic, suggesting that ablation of the N-terminal region of C protein doesn't influences the SRX of myosin. So then we, in collaboration with Dr. Previous and Dr. Washa at the University of Vermont, we determined the fast actin velocity, sliding velocity through the C zone in the absence of C0, C1F region compared to the control where we have the full-length myosin binding protein C. You can see that there is a two-phase of sliding velocity when we have the full-length C protein compared to the transgenic protein. So we can explain this in this schematic diagram. So in the top panel, you can see that we have the normal sarcomere with the full-length C protein. When we slide actin through this, it goes faster in the absence of C protein. And when we have the C protein, there is a slow in phase, and then it goes off. And when you compare that with the N-terminal ablation, you can see that there is no slowdown phase in the absence of C0, C1F. So that clearly demonstrates that the N-terminal region plays a role in terms of interacting with actin and regulating the contractility. So then we also collaborated with Dr. Tom Irving and Dr. Wei-King Mong at the IIT in Chicago to determine whether loss of C0, C1F region alters the distribution of cross-bridge mass between thick and thin filaments. We performed X-ray diffraction studies using skinned papillary muscles under resting conditions. Again, here data showed that the 1:1:0 intensity ratio in the skinned papillary muscle from transgenic hearts was significantly lower, while the interfilament lattice space was significantly higher in transgenic compared to the non-transgenic myofibrils. So this data kind of suggests that loss of the N-terminal region leads to a net transfer of myosin heads or more mass of myosin heads closer to the thick filaments backbone with a greater interfilament spacing. So if you look at this full-length myosin binding protein C, clearly C0, C2 has more binding studies, which includes the complete M domain and as well as full C2 domain. These regions can bind to myosin S2 region. So also several studies, I just listed all of them are here, clearly shown that the sufficiency of C0, C2 to activate force production in the absence of calcium, and it can activate thin filaments. And also the C0, C2 is sufficient to properly localize in the sarcomere. Also, very recently, Julian Seltzer's lab showed that using adeno-associated virus gene transfer with the C0, C2 can regulate cardiac function in vivo and also can rescue the cardiomyopathy phenotype early age using the animal studies. So altogether, these data suggest that increased therapeutic potential of using the C0, C2 region for normalizing myofilament function during diseases. So we wanted to test further whether C0, C2 in our study can be used to restore the function that we see, altered function that we see in the transgenic myofibrils, and also improve the actomyosin interactions. So what we did was we used both transgenic as well as failing myofibrils that were permeabilized with C0C2 recombinant proteins, and then performed the P-calcium force measurements. You can see that by permeabilizing C0C2, it increases not only in just the transgenic, but also the non-transgenic, the significant increase in force of production, but also the failing also, there is an increase in the force, maximal force, as well as the rate of force redevelopment also significantly improved, both in transgenic and non-transgenic, and same here, non-failing and failing myofibrils across. Also, we performed the similar study using myosin binding proteins in null myofibrils. So we permeabilized the myofibrils with the C0C2, and then we measured the maximal force as well as the rate of redevelopment, and confirmed again that C0C2 is enough to restore thick and thin filament interactions and can improve the contractility. So with that, I would like to summarize our study, and we'll have a lot of time for a lot of discussion so that we can discuss additional points. So in summary, C protein regulatory action depends on the N-terminal region, and the deletion of the N-terminal region, C0C1F region, accelerates cardiac contractility as well as results in cardiomyopathy or heart failure. And administration of C0C2 to permeabilized cardiomyocytes from failing hearts restored steady state actomyosin interactions. And lastly, C0C2 of C protein augments contractility and thin filament interactions. So it's kind of a nice and timely study to report the important role of the N-terminal region of C protein. Our future studies include, we are now performing in vitro studies to define the specific amino acids that are involved in the C protein, myosin, and actin interactions that could impact the sarcomere contractility. And also, we are using the C0C1F protein or the region to define the pathogenic role to modulate inflammation in the post myocardial infarction conditions. With that, I would like to thank all my mentors and collaborators from my PhD to all the way to today, my postdoc, my former and present lab members, and all the funding agencies. I hope to see you all in person at the ISHR North American section on September 12th in Denver. And so I thank you for your attention and happy to take your questions and comments.
Thank you, Sakti. That was a wonderful presentation. Really, really enjoyed that. And we do have questions that are beginning to come in. So let's see. I see some in the chat, but we'll start with question and answer, and I'll return to the chat. So Anant Chopra says, great presentation, big fan. There are some truncating mutations in myosin binding protein C that lead to hypercontractility. How does this manifest itself in the workings of myosin-mediated force generation?
So again, we all know 70% of the C protein mutations are the C-terminal truncations. So when you remove the C-terminus, protein is, unable to localize in the sarcomere, because the C-terminus is essential for the C protein's localization. So what happens in a normal condition in a myocardium, when you have the mutant protein lacking C zone, it's removed by the UPS system, and so that the N-terminal portion is never been available in the sarcomere. So again, we always see the haploinsufficiency, or the if you have disease condition, then maybe these can accumulate, but over the time it can randomly binds in the sarcomere. That's what we see when you overexpress C0, C1F, it can cause severe heart failure phenotype.
Okay, and finishing up the question and answer. Oh, wait. Lucy Carrier asked, hi, Lucy. Thank you for your beautiful presentation. What can you do to confirm that the effect on cardiac function is due to your N-terminal fragment and not reduced level of endogenous C protein in the transgenic mice? I suppose that.
Yeah, so in the transgenic, we have 80% the transgenic lacking the N-terminal, and then 20%. Again, we know, every 43 nanometer, there is 3 myosin binding protein C with a perfect ratios. So in the phenotype we observed is definitely due to this 80% incorporation, right? The lack of the N-terminal region. So I think that's what we are seeing. So again, when the sliding experiment clearly showed that, I think in the supplement data we showed like 10% we do see there is a 2-phase velocity, but 80-90% are single velocity. There is no 2-phase. So having the 80% knockdown of the N-terminus, the phenotype is clear that they develop hypercontraction, they develop increased rate of relaxation, and then eventually develop heart failure.
Okay, going to the chat. Michelle, and I apologize for the probable mispronunciation, but Michelle Parvatir says hello to us. And then she asks, let's see, in relation to the studies you showed overexpressing the 40 kD myosin binding protein C fragment in mice, did you look for the presence of autoantibodies against the fragment in mice with heart failure?
It's a really nice question, Michelle. Thank you. So again, these were developed by Robin's lab at Cincinnati Children's. So after I moved to UC, I tried to get that access, but the models are not available to us. So I'm hoping to develop such a model. Definitely, I think because when we overexpress, the protein is pretty much all in the sarcomeres. There should be some leak. There should be some release from the myocytes. So definitely there is a chance, but I don't have data.
Sam Dudley says, Sakti, wonderful talk. What happened to diastolic function in the N-terminal deletion?
Thanks, Sam. Diastolic function, because we do see, we didn't see very clear differences. Probably we didn't look very early on in the study. We did starting from 3 months. Then at that time, we did not see any diastolic dysfunctions. I mean, we do see the systolic dysfunction, but not diastolic. Probably it's too late, but we have not started, we have not looked at the early stage. So there, yeah.
Okay.
I don't know whether it can affect diastolic function in these mice.
Okay. Keithjones says, very nice study, Sakti. I wonder if you or anyone has put C0C1 into skeletal muscle fibers to see if it interacts with the skeletal muscle myosin and changes dynamics.
That's a really good question. I never even thought. Again, these are interesting thing to do. Again, when we propose this as a grant proposal, people will ask, okay, what's the biological significance of doing it, right? So if cardiac protein is expressed in the skeletal muscle, this could be really a potential experiment to try, but as of now, we don't see that. So, but hypothetically, we can test this in skeletal muscle.
Okay, Alicia Matias asks or says, very nice talk. It's, is it known whether beta-adrenergic stimulation phosphorylates these specific residues? And if so, what is the consequence?
Yeah, so the phosphorylation motifs are 273, 282, 302, and even Sam Harris, she reported 307 site. There are multiple sites in the M domain. So these domains can be phosphorylated by various kinases, including PKA, which is one of the beta-adrenergic stimulation-based kinases. So what happens is when these sites are highly phosphorylated, so under normal condition, myosin binding protein C is 60 to 70% phosphorylated. So when we have hyperphosphorylation means 100% fully phosphorylated all the sites by one of these kinases. So under this condition, we do see the literature reported that increased contractility and then the rate of force redevelopment also increase, meaning that more cross bridges, more increased force generation. Because it's kind of removing the brake from myosin head. So myosin can now, can do more interaction with the actin and generating more force.
So this is exciting. There's lots of questions, just so the audience knows. I'm trying to do things kind of in the chronological order that the questions are popping up. So just be patient and we'll get to all of them.
Time, so.
Yeah, and which is one of the great things of Zoom is everyone can write their questions in here. But okay, let's see. The next question I have is from, again, I'm not good with these names, Rajarajan Tandavarian. Great talk. Is there a difference between right ventricle and left ventricle?
Really good question. Again, based on the literature, we do not expect anything. Again, no, I don't think there is a systematic study defining the expression profile of C protein between left and right. I don't think, so I can't really comment on that, but I don't also expect to see a big difference. So maybe Sam can answer this question.
So we're starting to do some studies, and your former postdoc, Dave Bearfield, I think has looked at C protein expression in the different chambers. And I don't think there's a difference in the ventricles, but there is in the atria versus ventricles, I believe. But okay, so let's see. Next question, Rishita Bakchi. What kind of effect would the N-terminal deletion have in diabetic mice?
So in diabetic, you have already diastolic dysfunction, kind of heart failure with preserved ejection fraction. Probably it can be beneficial. I'm not sure. Again, the level of the N-terminal deletion, if you can manipulate, mild to moderate level, maybe that's beneficial because it can help to improve contractility. That's all I can predict. But again, diabetic can lead to develop diabetic-induced cardiomyopathy and develop heart failure down the road. But I know early on that they have diastolic dysfunction. So maybe having kind of mild to moderate level of the N-terminal deletion can be beneficial.
Interesting idea. Okay, from Don Burrs, great talk, Sakti. C0C2 seems to stabilize the actomyosin interaction and thereby increase force. Does this enhanced actomyosin interaction slow relaxation? How does C0C2 enhance myofilament calcium sensitivity? And how does that interact with the direct actomyosin C0 complex? Yeah. Lastly, 3, how does phosphorylation alter thin filament interaction of C0C2? So I can remind you each part of these.
Okay, so first question, thank you so much, Don. The first question is what?
Does this enhanced actomyosin interaction slow relaxation?
Yes, so it definitely, because the C0C2 has binding sites for both myosin and actin. So definitely it does that. Relaxation, I think so, because of the phosphorylation motifs are included, and then also myosin binding S2 regions also included. So definitely I believe, but again, I don't have a data on my top of my head how much it can involve in controlling the relaxation, but I can expect it should.
Okay, how does C0, C2 enhance biofilament calcium sensitivity?
Yeah, so there are a lot of studies that have shown that the C0, C2 can modulate the changes in the thin filament activation. So one of the things in the N-terminal region is mimicking calcium. So that's the kind of the new theory last few years evolved, that the N-terminal region of C protein act like a calcium. It can compensate the lack of calcium kind of concentration in the C zone region compared to the closer one to the Z zone. So just the N-terminal region of the protein is enough to activate thin filaments without even any calcium. So through several mechanisms, one is that it can change the alpha trophomyosin structure in a way that it can change then thin filament activation, so that can modulate the contractility. So it does so.
Okay.
Third question.
So yeah, and let's see. Third question, how does phosphorylation alter the thin filament interaction with C0, C2?
Yeah, so again, a lot of studies, including Dr. Harris. So phosphorylation moves away, and it has also actin binding. There are two actin bindings. One is phosphorylation dependent, another one is phosphorylation independent. Again, so these phosphorylation independent and dependent bindings can affect thin filaments interactions and then changes the binding affinity of calcium.
Okay. Okay, next question from Hadi Khalil. Great presentation, Sakti. What happens in terms of the cardiomyocyte size? Do you see any interstitial fibrosis in the transgenic mice?
Yes, we do. Yes, we do. We have reported that in the supplement data. The transgenic mice expressing the truncated N-terminal region show hypertrophic phenotype with fibrosis. Yes.
Okay. Another question comes in. Great talk. A semi-technical question. How does recombinant C0, C2 incorporate with the rest of C protein impermeabilized cardiac fibers in vitro?
Sorry, would you repeat the question one more time?
Sure.
I don't want to look at when I look at the chart, then I forget to pay attention to the question. Sorry.
It's okay. So how does recombinant C0, C2 incorporate with endogenous C protein impermeabilized cardiac fibers in vitro?
Yeah, so the permeabilized system, you are removing all the all the cell membranes. So you can permeabilize, you can exchange sarcomeric proteins, even troponins. You can easily exchange everything. So when you permeabilize the recombinant proteins, these recombinant proteins can get into the sarcomere. But the question is where they go, how they bind, right? So all we know that the C0C1F or C0C2, they go into the sarcomere and they bind throughout the sarcomere wherever there are binding affinities. For example, the C0C2 has both actin and myosin, so they can go wherever the actin and myosin binding sites are available for them, they attach. So we can't control that. So that's what this system is.
Okay, Thomas Lynch had a comment in response to Lucy's question, and so I think it's worth pointing it out. Yeah. So Thomas says, Lucy, the transgenic mouse was made on a non-transgenic background, so there's still 20% wild type C protein present. The total amount of C protein in the mice is consistent between non-transgenic and transgenic, so the full length and truncated combined. So the phenotype is likely due to the ablation of the N-terminal region.
Thanks, Tom. Thanks for joining.
Let's see. So Sarah Schumacher-Boss has, hi Sakti, when you have a shift to all fast sliding and no fast and slow phases, does this alter the metabolic demand or mitochondrial performance in the myocytes?
Of course, yes. So when you remove that, then you have increase in force production, meaning that you need more ATP, right? So there will be more demand of energy. So mitochondria should be, very active and generating more, utilizing more energy to generate more ATP. So probably that are the reason that these mice develop phenotype as a compensation.
Okay. Christopher Solis asks or states, great talk. What could be C0, C2 influencing more? Activation of the thin filament or the myosin SRX and DRX populations?
I think, again, this should be really dissected at the molecular level. All we know right now, I mean, these are in vitro studies looking at specific, parameters. We know it can activate thin filaments, but in terms of SRX, I am not sure. Probably it has the binding sites with myosin S2. So maybe the phosphorylation and dephosphorylation of the M domain can influence the SRX of myosin. So there is a possibility, but I think we have to test that.
Okay. Viswanathan Rajagopalan. Thanks for another great talk, Sakti. Could you say anything about C protein's interactions with diverse non-coding RNAs?
Oh my God, I never even thought about that.
That's a tough one.
Yes, that's a tough one. But again, it's possible because again, I showed like C protein is one of the predominant protein in the sarcomere. So it should be tested.
Okay. And Lucy Carrier is clarifying her question again to you and Thomas Lynch.
Yes.
She points out that she realizes that it's on the non-transgenic background, but the 20% of normal C protein per se could have an effect. Therefore, it would be good to prove that this N-terminal deleted fragment affects the contraction all by itself.
Yeah, we agree, Lucy. Thank you again for your question. When we move from transgenic to knockout background, we can study the 100% impact of the N-terminal deletion. Again, it's all detailed experiments, but what we see, even the Mike previous really showed very nice that he does see that the 20% influences the sliding velocity of actin. So the 20% has some contribution. Yes, we do see that. However, the 80% of the truncation is enough to show the functional differences in terms of single cell level, in terms of cardiac level. So I'll be sure that in this study, the 80% expression of the N-terminal truncation is sufficient to cause or to show the importance of the N-terminal region. So again, adding the 20%, yes, it's another addition of experiments. It takes more time for graduate students to get graduated. I'm glad Tom took even more time to get this completed. So thank you, Lucy. Thank you so much for joining from Germany. And we look forward to seeing you next year at the ISHR meeting, hopefully.
So just for completion, Thomas Lynch actually responded in the chat and said virtually what you said, Sakti. So you guys are simpatico on that. So Charles Chung, hi Charles. Sakti, very much appreciate the nice history background and the very thorough study of myosin protein C. Regarding the X-ray data, do you think that myosin is actually going towards the thin filament, or could loss of mass from the C0, C2 account for the difference? Ooh, that's good. Or could the truncated C protein be binding to the thick filament?
Yeah, so I think the truncated C protein binds to myosin predominantly because it has the C2 partial M domain and C2. That's what it's more towards myosin. So it's not reaching the thin filaments. That's exactly the point we are making. You have the M domain, you have the C2 domain, so it goes to myosin. That's what the SRX data show, that there is no difference in SRX of the myosin as well. Even though we do see, the heart failure phenotype, pretty much all the truncated protein moves to myosin.
Okay.
Thank you.
Let's see. Next is Helena Viola. Hi, Sakti. Thank you for your talk. Does this interaction play a role in the development of hypertrophic cardiomyopathy, predilation failure? Many thanks.
Hey, thank you for joining from Australia. This is a big time difference. Again, 70% of the myosin binding protein C mutations are the C-terminal truncations. Yes, there is no way you can, overexpress or regulate the N-terminal alone. Again, when you overexpress C0, C2, it's going to be all over in the sarcomere. It's not going to be only in the C2 or C zone. So that's one thing we really have to look very carefully. And so several myosin binding protein C mutations that are in the N-terminals at the phosphorylation motifs, they can do the similar job, ablating the binding between actins or the myosin S2 can mimic this kind of phenotype. Again, there is no systematic study, but you can predict that based on the binding interaction studies. But these are the systematic studies we have to do down the road. Right now, there is none.
Okay, it seems like C0, C2 has some therapeutic potential in vitro. Should this potential be demonstrated in vivo? How would you translate it from bench side to bed?
Yeah, so this is a really good question. So Julian Seltzer, he from Case Western from Cleveland, they already showed a paper that using adeno-associated virus expressing C0, C2 can rescue or partially rescue the hypertrophic phenotype in their null mice. So what they did was they treated the null mice early on, embryonically, starting from embryonic stage. They show there is a partial rescue. So there is a potential. Again, we don't know the long-term impact. We don't know whether it can be very specific. So again, we need a systematic studies, and these are the limitations they acknowledged in their paper as well. So maybe timingly overexpression by adenovirus or by gene therapies can help, but this has to be done down the road. And Julian, he has a pattern on the C0, C2 just for therapeutic purpose.
So Sakti, I'll jump in here with one question because it's related to it that I've been thinking about. So it seems like C0, C2, and this actually circles back to Don Burz's question, that C0, C2 in my hands will have an activating effect even without calcium. And so it can increase the passive tension. So it would be, you'd predict that it would affect diastole and relaxation. So is this something that is of a concern when you're thinking about using this therapeutically?
Yeah, so, I mean, again, therapeutically, you can modulate the level of expression. If you throw 100%, yes, it can influence us. But if you can modulate the level of expression that is sufficient just to activate only the thin filaments, or just to only influences the relaxation, it should be beneficial. Again, the time of modulation, the level of modulation should be established. But again, based on all the in vitro and in vivo or in vitro studies show that it can be done. I mean, it does influencing the thin filament activation, as well as myosin are activating the myosin function as well.
Okay, great. And we are over time, but there are two more questions. So everyone who can stay, we've got two more good ones for you guys. Okay, Somebody asked, do you believe there might be an advantage in using myosin-binding protein C3 gene therapy for the treatment of myosin-binding protein C-associated HCM using mutant C-protein cDNA, which cannot be cleaved?
Thank you for the question. Again, so Lucy Carey's group in Germany has a lot of work on like gene therapy using the full-length myosin-binding protein C or the phosphomimetic. So it is a potential because if the mutant proteins are protected against calpain cleavage, it is really beneficial. Again, you know, even the C0, C2, you can modify the M domains with the calpain protection. So it can be used, but again, this has to be tested. Again, I was very interested in trying these kind of experiments, but after seeing the you know, initiation by Dr. Lucy Carey as well as Julian Seltzer, I kind of slowed down. I divert myself in a different direction because they're already in this similar area of research.
Great, thank you. Palin Kumar Manoharan says, fantastic presentation, Dr. Sakti, very informative. Does vaccination against C0C1F prevent initiation of inflammation, signaling, and cardiac remodeling post-MI? Great question.
Thank you, Palani. Yeah, I think that's one of the kind of interesting, you know, so I am working with Dr. Sumant Prabhu on submitting on R01 on how this C0C1F influences pro-inflammation or activation of pro-inflammatory markers. And one of the idea is that, you know, if we use antibodies against C0C1F post-MI, can improve or can prevent the inflammation activation. So there is a potential, but again, we have to test all this thing down the road. Again, the vaccination is another great idea. But again, if we see C0C1F is a great cause of generating inflammation, it's a path to go.
Okay, well, the questions have tapered off, and actually, Sakti, I could continue talking to you all afternoon because I've got more questions, but I think in the interest of time, I'll just table them at the moment and say what fun this was. Fantastic presentation.
Yeah, I'm glad
Thank
I was
you very
able to
much.
do it early on to have enough time for all these questions.
Yeah, so that was great, and thank you so much for the audience participation. This is wonderful coming out of the pandemic, hopefully, and getting to see so many names of colleagues that it's been too long that we've connected with. So I thank the audience for everything, but thank you, Sakti.
Thank you, Sam. Thank you so much. Thank you, Ron. And thank you all for joining. It's just a nice platform for all of us to connect. And hopefully we all see at the ISHR meeting in Denver very soon.
Okay. Thanks, everyone. Bye. So Ron, are you saving the chat so that